Well, I thank you. I was asked today to speak about the challenges that we have with next-gen sequencing, and specifically in the validation. And I'll take a second and just describe in my brief wording, analytical validation of an assay is basically an exercise that clinical labs go through to demonstrate that the assay is actually measuring what it should be measuring and that you can do it reproducibly. And as you can imagine, we've got quite a challenge when we come to next genome. A quick disclosure, I have nothing to disclose other than any opinion that I'll give you is mine and not representative of the government or my employer, Leidos Biomedical. Next generation sequencing, as you've been hearing, it's an extremely powerful assay technology. People refer to it as multi-analyte. It's really giga-analyte, where some people are sequencing the entire genome, 3 billion plus bases. So that's a lot of analytes, and to be able to demonstrate that you can do this accurately is a daunting task. It's obviously a very complex system, because not only do we have the wet lab molecular biology of taking a DNA strand and running enzymes down it that will capture the sequence, but then after we've captured the sequence on the instrument, it needs to go into a computer with algorithms that will actually then go and take all of these pieces of DNA that we've generated, the signals, map them back to the genome, and then go in and look for variants that are the unique somatic mutations that occur in the cancer. These algorithms are extremely complex, and often they're black box. You basically are taking some trust in the fact that it's doing everything that it should be doing accurately. There's 3 types of next-gen sequencing that are being used right now and used in clinical labs. As Mia was talking about, we have a lot of people that are using targeted next-gen sequencing, and that's where it's a subset of genes that have been defined as worthy to be sequenced and of value to understand as far as working up a cancer patient's somatic mutations. A lot of labs now have begun to do whole exome, and this is the next scale, basically looking at every single coding region of all of the genes in the genome. And then the most complex, obviously, is the giga-analyte, where you're sequencing an entire genome. As you can imagine, and as you've seen, the technology is improving rapidly. So that's one of the major challenges we have in a clinical lab, is when do you lock down and say, okay, any new improvements to this platform, I'm going to leave behind because I need to have reproducible results as I go forward. And so therefore you're stepping away from the improvements. So as far as steps to validating, and this is more or less the way that we've done it, I'll give you an example as we go on in this talk of how we're doing it to support the NCI MATCH clinical study, which is set to launch sometime in February of next year. But basically, one of the things that you should do at the very beginning is define the intended use. How is the assay going to be used? What is it going to report? What is a clinician going to do with this information? Because that will help you actually then decide what it is that you need to demonstrate in the analytical validation is reproducible. The assay is a system, obviously. We're taking biopsies from a patient, and understanding how that biopsy is handled is extremely important to the end results that one gets out of the assay. We've engaged the FDA, actually, because all of our assays that we're using to support clinical studies are going to be used to select treatments for cancer patients. So rather than going off and doing this, we've gone to the FDA to have pre-submission discussions to find out what they can do as far as giving us advice on how to build the assay and how to actually go through the validation to be certain that we're doing the best job that we can. We also take a period of time with the assay to do what we call feasibility testing, and that's at the very beginning, just to be certain that you understand how the assay works, what the strengths are of the assay, and more importantly, actually, is understanding where the assay has weaknesses so you can mitigate. And then you can move into the actual analytical validation phase of testing assay performance. So the intended use, there's a couple of different types of assay intended use, and one would be pure clinical research. If you're going to get specimens and then just sequence them for the pure sake of research to see what you can discover. Another approach is an integrated assay, which the assay is actually going to be embedded into a clinical study, but the results are not going to be used for patient management. Again, this is a type of assay where you're collecting biopsies specifically to run the assay, so you should be putting time into analytical validation in that type of scenario. And then the NCI match study, the assay that we'll be using, it is considered an integral assay. Basically, we're collecting patient specimens. They will be sequenced, and the results of that sequencing then will help us select the treatment and enroll the patient into the study. And obviously, this is where you really need to be certain that the assay is performing acceptably. As I mentioned, assays are systems. They begin with the collection of the biospecimen, and they actually end with the reporting of the results. So every step in between should be known, it should be locked, it should be documented that you can get reproducible results. As I've already mentioned, next-gen sequencing assays are very complex, and once you've locked these parameters, if you deviate, you're in for potential of changing the assay's performance criteria. So it's very critical once you've defined the system to lock it and keep it locked, even if improvements come along that could potentially make the assay better. What I want to really stress is each one of these assays is very unique. There's all types of parameters, there's all types of tweaks and changes that one can make as far as just the analysis of the data, how one sets the filters on whether or not you will accept anything that has less than 20-fold sequencing depth or greater than 30-fold. All of these are different parameters that will impact the overall results. So you really need to lock it, and you really need to understand that each one of these assays is unique. I would say, and I will talk a little bit about it later, that we really need reference materials so that we can begin to share these across different laboratories to find out how these assays really do compare. And I'll leave it at that. The NCI match study is going to be, I guess, the basket of all basket studies. It's a multi-arm basket study. So it right now has about 22 different treatment arms. And each one of those treatment arms will have a drug matched to somatic mutations, and it will go across all different tumor types. So again, it's a collection of different treatment arms all together in one basic IND. The assay itself will identify mutations, gene amplifications, and gene fusions in patient tumors. And each one of those different types of variants will be used for different treatment selections. The assay, again, will be used to assign the patient to the relevant treatment agent arm. And we're going to use a rules-based approach. In other words, this isn't going to be an assay that goes to a clinician that then will have a tumor board or a group of pathologists and clinicians, molecular biologists that will make a decision on how to treat the patient. It will a priori have rules that will say these mutations are matched with these treatments, and if we have more than one mutation, how we deal with that to actually get down to a final treatment. Because we're doing this, we're going to be screening a large number of tumors, and we wanted to have a large number of treatments and mutations so that we had the best opportunity to get as many patients that donate a biopsy to actually be enrolled. The basic schema for the study is we'll begin with the next-gen sequencing. If an actionable mutation is detected, the patient can be enrolled, and then again, we'll use a rules-based to actually select the arm that the patient will go on. They have the opportunity, if they progress and they have another variant, to actually go back on to another treatment arm. They also will get a biopsy at the time of progression. So the actual match assay that we're developing, again, I've already mentioned it's an integral assay, and its intended use is to screen patients for entrance and also to select the treatments. One of the interesting things that we decided to do in this study is to select 4 clinical labs that will run the exact same assay. So we've got MD Anderson with Stan Hamilton and his group, Yale, Jeffrey Sklar, and MGH, John Iafredi, and then a lab at Frederick National Lab for Cancer Research. We're all going to run the same assay, use the same SOPs, And part of our validation will actually be demonstrating that we get the same sequencing results in each one of the 4 labs. We're using levels of evidence, as Mia spoke about, and we're using those level of evidence to help us to select the treatments that would be matched to the appropriate mutations. There'll be a rules-based engine running in a device called Matchbox, which will actually apply those rules based on the sequencing results to select treatment. We've already consulted the FDA, had a pre-submission discussion, shared our validation plan, got some very good, insightful information, and revised the validation plan based on those discussions. And now we're at the point of moving forward. The actual assay system and the workflow, it starts again with the biopsy. We'll collect 4 core needle biopsies from the patients. They'll be shipped overnight in formalin to MD Anderson. And MD Anderson will be a central pre-analytic site. So they'll do all of the processing. They'll do enrichment, actually, because we're aiming to get 70% or greater tumor content in each one of the samples that get sequenced. After the sample has been processed, nucleic acids then will be distributed to one of the 4 labs. The sequencing will be done, again, using locked SOPs, and the results will go into Matchbox for treatment selection. We'll collect all of this data in a clinical database. So we've just finished up the feasibility testing, and I can say the assay is performing very well, but we did find some weaknesses. We identified the fact that we were having some weakness with copy number variants, that each site had a little bit different baseline, and we're in the process of mitigating that right now. But we have had the opportunity by running this feasibility to actually get to the point where we've locked the SOPs, and we've developed quality checks where basically we know what we're expecting the assay to tell us throughout the different stages. And now that we've locked with these quality checks, we know when a sample is not performing up to snuff, and it would actually be flagged and repeated if possible. As far as a meeting with the FDA, we had that meeting. We actually are reporting on 4,048 known variants. So the assay is fairly large as far as what the report will read. And as a result, it's clear that we can't validate every single one of these analytes. So what we've done basically is chosen a group of the analytes to represent each of the different variant classes. And we chose those based on what we consider to be highly frequent, ones that we know we'll see a lot of, as well as ones that we know will be difficult to sequence. And that's what we'll use for our analytical performance testing. So moving right along, the different types of variants, we have small nucleotide variants, indels, large indels, copy number variations, and gene fusions. So we'll test each one of these types of variants. Specificity is very important, and we actually consider the specificity to be more important than the sensitivity. In other words, we would prefer not to put a patient on protocol that would be a false positive. So we're erring to have high specificity at the expense of potentially missing a variant that would get a patient on the study. I will tell you the sensitivity looks promising, but the specificity is where our focus will be. In validation, obviously, we also need to address the accuracy of the assay and compare it to other known assays, clinical grade assays, to be certain that the variants that our assay is detecting are actually there. We'll do our traditional repeatability and reproducibility, as I mentioned. This is a difficult study because we have 4 laboratories that we need to demonstrate reproducibility across. And then at the very end, we'll do what we call a fit for purpose. We'll run specimens that are collected at MD Anderson all the way through the pipeline, through the shipping, through the variant calling, through the Matchbox, to be certain that everything is performing where it needs to be in order to actually launch the study. So that's NCI Match and the assay that we're building for it. I wanted to just leave with some final thoughts. And as I started, I told you that next-gen sequencing assays are very complex. Everybody's doing it now. Virtually every clinical lab, every cancer center is doing next-gen sequencing. But we really don't know if we're getting the same results because we really need, an urgent need really, for reference materials I'll put a plug in for NIST. They have a genomes in the bottle activity that's going on, and that is to provide a certified reference human genome where we know truth. Everybody can sequence this, and they will basically be able to tell you that if you get an A at this position, your assay is accurate. We also, in my mind, believe need to start collecting in the microarray era. We had a convention called Miami, which was a minimal amount of information that need to be submitted along with any data set for publication. Right now, there's so many different parameters in next-gen sequencing. It would be wonderful if our publications begin to actually accept the sufficient information so somebody could reproduce the assay and get the same results if needed. There's just way too many parameters right now that if they aren't collected, we'll never be able to know how they did it. The other thing I should say is moving into clinical studies, which most of our activity is, you need to begin to plan for success and demonstrate clinical validity. That's beyond the analytical performance now of the assay, that we actually are detecting variants that are matched to a drug that show promise for treatment. And in order to do that, you need to start thinking about clinical utility, because that's the big hurdle that everybody has at the end of the day in order to get these assays out to where they can really begin to help patients with treatments. And I think there's a need for guidelines for clinically actionable variants. And I think David Solit will give us some background on what some of the activities in this area are. But as you saw, we all have these panels right now. We have a large group of variants, some of which we all agree are clinically actionable. But again, having some sort of reference standard that we can go to actually begin to match treatments with levels of evidence for patients would be very, very important. And I think we're in a time right now that everything is moving so rapidly. We have a great opportunity to work across different pharmaceutical companies' interests, to work across different diagnostic companies' interests, and in the patient's interest, to work collaboratively to be certain that we're bringing these assays up into the clinic so at the end of the day we do no harm, but actually really push the field forward. So with that, I'll sit down and turn it over to the next speaker.
Okay, we'll move to circulating biomarkers, and Matthias Holdhoff from Johns Hopkins will talk about DNA, tumor DNA circulating biomarkers.